science lovers. So today we are at the 8th Congress of Microbiology organised by the FEMS and the Spanish Society of Microbiology. And I thought, what better to do here than to talk to a few people that are presenting some posters and ask them to do us a summary in around 1-minute videos so that we can learn all about what their research is about and how they have presented it at this Congress. So I hope you enjoy this video.
Hello, I'm Christian, I'm from Germany. We are interested in anaerobic digestion, and here on the FEMS I'm presenting a description of a pre-treatment stage to produce acids for anaerobic digestion, for methane production. And we managed to make the very first multi-omics approach with a separated acidification stage. We use this to compare on a metabolic level the activity in mesophilic and thermophilic stages, and we compare the proteomic data with the microbial behavior based on 16S rDNA sequencing. And then additionally we were feeding the produced acids into a methane stage to convert them into methane. And we were investigating the microbial dynamics in relation to the microbial input from the acidification stage. And we proposed several interesting strains for microbial groups as marker for efficiency of biogas production, such as actinobacteria, methanocoleus or metanocyta.
Hello, I'm William Kapucha-Popovic from Faculty of Science and Split Croatia. And here I present my work on bladder cancer. We study bladder cancer, which is the 9th most common malignant disease globally. And in particular, we wanted to see whether bacteria that live inside this bladder contribute to the development of bladder cancer. And therefore, we collected urine samples from healthy people as well as from people that are diagnosed with bladder cancer. We isolated DNA, and we used DNA analysis to describe microbial communities that live in the bladder. And the result of our work showed that there are certain bacterial species. They're shown in this picture. There are several bacteria that are more abundant, that are enriched in bladder cancer samples. One of the bacteria, Fusobacterium nucleatum, particularly draw our attention because if this bacteria has already been associated with colorectal cancer, and in the future we would like to continue our work to see whether this applies to bladder cancer as well.
Hi, my name is Maria Junkova. I work at the Australian Centre for Ecogenomics, and my poster is about a very interesting topic, which is dealing with the dark matter, bacterial dark matter and viral dark matter. It means that we are studying the relations between the bacteria and viruses that can't be cultured. They are completely unknown, and we just can't get their genome sequences. So we can actually find these relations based on their genome sequence, but it's better to have like a real laboratory proof that it's working. So in my approach, I'm staining the viruses with the DNA and RNA stains, and then I combine them with the bacteria from the same fecal sample. And when the viruses take the bacteria, they become fluorescent. So I can collect them and I can sequence this pair of the viruses and the bacteria. And in this way, I can create something kind of network, allowing us to detect which viruses can attack which bacteria and whether they have different hosts. Because up to now, like all the scientists think that one virus can have just one bacterial genus as a host, but I think that I'm discovering that they can be more.
Good afternoon, I'm Pedro. I'm working at the Public University of Navarra and I'm doing my PhD there. and I'm going to talk to you about the project I'm trying to achieve or developing there. What I'm doing is to try to improve the capabilities of a bacteria, able to degrade one contaminant compound by making it produce biofilm. What is a biofilm? A biofilm is some of the bacteria that came together because they produce a substance like a glue that keeps them together and makes them more resistant and hopefully more catalytic ones or something. And I don't know, we tried different strategies, we choose the one that worked the best. We have tried different strategies, but we have to do some more trials to see which conformation works the best. And I don't know, I'll tell you later.
Hello everybody, my name is Ángeles Hueso Gil and I work in CMB. Our work focuses on Pseudomonas putida, and we are trying to build a surface reporter. So in order we can detect with a signal when Pseudomonas is attached to a surface. We tried at the beginning with the wasp complex, which in aeruginosa can be sensed in the surface and translating it into a cyclic DGMP signal, which is the main secondary metabolite that controls the lifestyle. And later we tried to build a reporter based on the prelate promoter of Pseudomonas aeruginosa 2. It seems to work well, but we want something that has a lower basal level. That's why we made a transcriptomic analysis comparing Pseudomonas putida attached to a surface. So that means cultured in a solid plate. And we compare it to a Pseudomonas putida cultured in a liquid media. And we saw different genes that were differentially expressed. Some of them were from categories that they were quite descriptive, but we are not interested on them. So that's why I have this list here. And these are the genes related to biofilm, cyclic DGMP, and motility related to. So these are the genes in which we are more interested. And we found some nice differences at 6, 12 and 24 hours. And we hope to have a better candidate for the cyclic DGMP reporter construction.
Hi, my name is Dani and I'm working at the Institute for Integrative Systems Biology at Valencia. And what is the major point of my poster? Okay, my poster is to try to unveil the relationship with the chain variation of the microbiota over time and its relatedness with the health and disease system, health and disease state of a person. Okay, so we can model the microbiota with Taylor's law, and that Taylor's law, which is an ecological law well known in the ecological world, can translate the fluctuations over time into two parameters that are V and beta, which V is the most important parameter, which is related with the size of the fluctuations over time. Okay, so a major V means that the microbiota is fluctuating over time more than a lesser V. Okay, so with this information we can place the microbiotas in a Taylor parameter space, and we can see that the microbiotas are really widespread in terms of variability, and that variability is also related with this equation. Okay, this equation is the Langevin equation, which translates the position of a bacteria over time with two main parameters, with deterministic mechanisms and the fluctuations. The fluctuations are related with the Taylor's law, and the deterministic mechanisms you have to calculate it. So solving this differential equation, you can have two different phases. Okay, you can have an unhealthy phase or you can have a healthy phase, which are the unstable or stable phases. So if you are in a stable phase, you are healthy, and if you are in an unstable phase, you are unhealthy. You can travel between both phases. For example, this person had two antibiotic intakes, or you can, for example, improve over time. So the variability over time is related with the health and disease of the persons in terms of the microbial populations. Thank you.
Hi, my name is Carolina and I'm studying in the University of Costa Rica. I'm going to tell you right now about my work. First of all, everything started with the isolation and characterization of some fungi that were growing on some lithograph. This lithograph belongs to Bernard Romain Julien, an artist from the 19th century. And when we isolated and characterized these fungi, as a result, we have that there were 21 different fungi isolated, belonging to the genera, for example, Penicillium, Trichoderma, Cladosporium, and Aspergillus. 2 of the whole fungi in the collection couldn't be identified through DNA sequencing, so we did a morphological identification. Through the morphological identification, which is basically observe the fungi under a microscope, we realized that there were 2 new species. Finally, we did an essay to determine how good were they to degrade cellulose, the component of the paper. And more or less, the 80% of the collection gave a positive result in the essay. This last result is important because we can use the different fungi, especially the good ones producing the enzymes, to degrade wastes of the agro industry, a problem in Costa Rica in this moment.
I'm Jorge Vázquez. I am a PhD in the biotechnology program of Valencia University, and I am working with Metagenomics. Working with Metagenomics in the HIV infection. Well, maybe as you know, the HIV infection is an infection that attacks the immune system and depletes the cells that are in charge from keeping the microorganism under control. So what we observe is that actually, even with a good response to the treatment, because nowadays the treatment is impressive, it's quite good, people is still dying 10 years before than the average population. And one possible explanation for that is the microbiome. So we observed that using different omic technologies, that the microbiome from HIV-infected people is different from controls. They have different species and different functions. So this microbiome community is able to form a stable community which lacks the capacity for produce really important compounds such as the butyrate. The butyrate is really important because control the microbiome, control the immune system, and also fuels the enterocytes, which are cells for the intestine. So what we observe actually is we model a network in which we observe that the lack of this butyrate is related to the systemic to the inflammation, to the disease progression, and to systemic inflammation, and all the syndromes and illness that are related to this phenomenon.
Hello, my name is Kristie Villanova, and I work in a biotech company here in Valencia, the name of which is Darwin. So in Darwin, we are interested in analyzing microbial diversity and also in exploring the tools that we can extract from this community. So basically, this is our poster, and in this poster we want to highlight the 3 main pillars of the research and the services that we offer in Darwin. The first one is that we mainly focus in analyzing unusual ecosystems. There are many reasons for that, but the main one is that they can be sources for new types of microorganisms with a lot of interesting applications. So for instance, we have analyzed the communities associated to solar panels, to coffee machines, or to dishwashers, because they have microorganisms and genes and enzymes that can be very useful because they are subjected to very extreme conditions, and this is very useful from the industrial point of view. Also, it's very important to highlight the kind of techniques that we use. We have a lot of experience in using multi-omic analysis. This is metagenomics, sequencing, proteomics, etc. But we think that it's very important to combine these multi-omic approaches with other type of technologies, mainly based in cultivation and in ecological characterization. And finally, we want also to explain the special focus that we have in cultivation techniques. And in this sense, we are developing new methodologies based in building metabolic networks, building heterogeneous media, in order to cultivate those microorganisms that are rare and that are very hard to control, but still are very interesting for us.